So now that we've talked about the cell cycle, we're going to start to talk about the different phases of cell division. So of course, we're going to start with interphase. And remember, this is a growth phase. This is not phase or part of mitosis. So when a cell is in interphase, remember that's the longest part of the cell cycle. A cell will spend about 95% of its time in interphase. And this is when it's going to do things like it's going to grow, it's going to develop more organelles, it's going to develop more cytosol, it's going to replicate its DNA, it's going to proofread that DNA, glue all of those fragments, Okazaki fragments and everything together so that we end up with two strands of DNA that are ready to proceed into mitosis, which of course is cell division in body cells of multicellular organisms. So when we take a look at our little graphic here, one of the hallmark characteristics of a cell in interphase, whether we're looking at a picture or we're looking at a micrograph like we are over here, is that we are always going to see the genetic material, the DNA, in chromatin form. So instead of seeing chromosomes all coiled up and ready to go in terms of dividing into other cells, we're actually going to have that long thread-like or noodle-like form of our DNA called chromatin. Now remember the nucleolus, which is kind of that dense area. So if we kind of compare cell to cell here, so over on the right here on our electron micrograph, this is what you'll commonly see when you're asked to interpret diagrams of cell division. This would be what I've outlined in red is a cell. So this is likely a plant cell because of its rectangular shape. And then this darkened area here is the nucleolus. And the nucleolus is responsible for ribosome formation. So while the nucleolus is essential in forming ribosomes for translation, it does not actually have genetic material within it. Okay, so the genetic material is restricted to the general nucleus itself. So again, back to our micrograph here, this would be the nuclear membrane. And then this would be a cell that we would consider to be in interphase because the nucleolus is very well defined, and you also have just this chromatin. You don't have any visible chromosomes that you can see doing anything at this point in time. Now, unlike those particular cells, this cell here clearly has something different going on. So this is going to be a phase of mitosis because you can see that we have this dark material here, which is actually chromosomes migrating to the poles of the cell. And then you can also kind of see that there's this really faint kind of line in the middle, which tells us that the cell is beginning to divide. Okay, so that is what we call a cell plate. And it is getting ready to divide that material and divide into two cells. So when we are thinking about interphase, just please keep in mind that this is not part of mitosis. Mitosis is specifically the cell division phase. And when a cell is in interphase, it's basically growing, doing what it's supposed to do as a cell, replicating its DNA, all in preparation for eventual mitosis. Now, if we take a look at cytokinesis in animal cells versus plant cells, there is a little bit of a difference. And just to recap, remember that cytokinesis means division of the cytoplasm. And for terminology purposes, remember that cytoplasm means not only the cytosol, which is that fluid part of the cell, but it's also the organelles. So the things like the mitochondria, the ribosomes, etc. So cytokinesis is the division of that cytoplasm among the two daughter cells. So when we have cytokinesis in animal cells, we have something called a cleavage furrow. So because animal cells do not have a cell wall and their cell membrane is a lot more, they don't have that rigid outer part, it's a lot more flexible, what we'll see is we'll have basically the sides of the cell will pinch in and we get like this furrow. And so think of that like, you know, when someone like frowns and they get like a furrow in their forehead. So we kind of have this cleavage furrow forming, and then the membrane is going to pinch in and eventually separate. Now, on the other hand, when we look at the plant cells, we have something that's much more structured, and we call this a cell plate. Okay, so you can see how we have this really kind of structured line across the two eventual daughter cells. And remember that the plant cells have that really rigid material, that structural material, cellulose, right, on their cell wall. And so the reason why this cell plate is also a lot more rigid looking is because we need development of cellulose to happen here, so that when we eventually do have our two daughter cells, we don't just have this kind of open kind of part of the cell without any cellulose, we actually have that full cell structure that we associate with a plant cell. So cleavage furrow and cell plate. So taking a look at just kind of the basics of mitosis, remember that once we're out of interphase and we launch into cell division in our body cells or our somatic cells, this is the process called mitosis. And one of the key things to remember about mitosis is that this is all about conservation of ploidy. So we're going to start with diploid and we're going to finish with diploid, okay? So we want the daughter cells to be genetically identical to the parents. And because there's no gametes or gamete formation or anything involved here, and because these daughter cells are genetically the same as the parent cells, we actually refer to this as asexual reproduction, okay, because there's no gametes involved. So as we said, this process of mitosis, because our daughter cells are genetic clones of the parent cell, we conserve ploidy, okay? So in this particular parent cell, if this were a human, we would have 46 in the original somatic cell, and then we'd have 46 chromosomes in the two daughter cells as well, okay? So we conserve ploidy in mitosis. So now we have to talk about the specific phases of mitosis. And one of the easiest ways to remember this is the mnemonic PMAT, or I guess it's more like an acronym, PMAT. So the P stands for prophase, the M stands for metaphase, the A stands for anaphase, and the T stands for telophase. So when we look at our first phase of mitosis, we call this prophase. And in prophase, this is where we're going to start to lose that thread-like chromatin DNA form, okay, that we had during interphase, and we are actually going to get that supercoiling happening, and that chromatin is going to coil, supercoil into chromosome form. Now, when we look at our kind of little abbreviation here, DS stands for double-stranded, okay? So that's one important thing about prophase. We have chromatin condensing into double-stranded chromosomal form, because remember, the chromatin itself at the end of the second growth phase was double-stranded. Okay, so if we take a look at our little graphic over here, you can actually see the chromosomes have the two connecting chromatids, right? And so we have double-stranded chromosomes. And as we learned last time, that little piece where the chromatids are bound together is called our centromere. And then the other thing that we have on here that is a little bit new is we have the aster, and the aster is this structure here that includes the centrioles, etc., that are, and centromeres, centrioles, which are necessary for cell division. And then the other thing you'll notice here is we have the beginning of these little structures called spindle fibers, and they kind of look like little spindles. So the other thing to remember in prophase is that the nuclear membrane and the nucleolus are intact, okay, because what's happening is we're getting that condensation of those chromatin into the double-stranded chromosome form. Eventually, as prophase continues, the nuclear membrane and the nucleolus are going to dissolve or disappear, and that's because we need those chromosomes to come out of that nucleus. The centrioles begin to migrate to opposite ends of the cell and create spindle fibers. So the centrioles are these little like cylindrical, they almost look like little cylinders, little structures there. And then the asters are the little structures that kind of radiate out from the centrioles. Now in our program of studies, asters are not, we don't need to know asters specifically, but the centrioles we do. But the asters, you can see the spindle fibers kind of form from the asters, okay? So the centrioles begin to migrate to the opposite poles or opposite ends of the cell in preparation for division. And then by definition, those little centrioles are little microtubules that help migrate and separate the chromosomes. So the next phase in our PMAT is metaphase, okay? So remember we had P for the prophase, M for the metaphase. And this one is very, very distinguishable because you can see what's happening here. So first of all, and importantly, the chromosomes are still double-stranded, okay? So you can see that here, right? We have the chromatids attached together, so they are still double-stranded chromosomes. And then what we actually call the middle part here where the dotted line is, where those chromosomes line up, we call it the metaphase plate. Sometimes it's also called the equator. So kind of like the Earth has the equator that runs on 0 degrees longitude, same type of idea. So the double-stranded chromosomes are going to line themselves up, and notice that they're attached to spindle fibers there, right? Okay, they aren't just like floating around randomly. The centromeres are connected to those spindle fibers. So during metaphase, we're going to have those double-stranded chromosomes lining up on that metaphase plate in preparation for division. And the other thing I want you to notice that you can see just a little bit subtly here is you can see that the little centromeres, you can see that they're kind of starting to pull apart. So the centromere is attached to that spindle fiber, and I bet you can probably predict what's going to happen. Half of that is going to go this way. So one sister chromatid is going to go to that pole, and the other sister chromatid is going to go to the other pole. So keys to remember with metaphase, we are still in double-stranded form. Those little centromeres attach to those spindle fibers and line up along that metaphase plate or that equator in preparation for the next phase of mitosis, which is anaphase. So P for prophase, M for metaphase, and A for anaphase. So just like I asked you to predict before, you can see that where those centromeres were attached to the spindle fibers, they are now being pulled to opposite ends of the cell. And notice what's really, really important here. Those centromeres have replicated so that each chromatid has its own centromere attached to a spindle fiber. And notice the form of the chromosomes now. We've gone from double-stranded in prophase and metaphase to single-stranded in anaphase. So that's key. So now what's happening is those single-stranded chromosomes are beginning to migrate to the opposite ends or opposite poles of the cell in preparation for cytokinesis. So our last phase before cytokinesis is telophase. So this is the T of the PMAT, right? So you can see what's happening here. So this is obviously an animal cell because we have a cleavage furrow happening as opposed to a cell plate. So you can see that the cell membrane is pinching in, right? Along that furrow, it's pinching in preparation for final division. And then the other thing is notice that we still have single-stranded, but notice what's happened here. The chromosomes are actually now starting to unwind and go back to chromatin, okay? Because remember, the only time we need actual chromosomes in our cells is during these phases of mitosis. So by the time we get to the end, we get to telophase, the single-stranded chromosomes are now starting to uncoil and go back to single-stranded chromatin form. And then the other thing that you should notice here is that the nuclear membrane and the nucleolus are starting to reform. So see these kind of little structures here, right? This is the reformation of the nuclear membrane or nuclear envelope, and this is the reformation of our little nucleolus in there. So notice that our cells are kind of going back to quote unquote normal, and then once cytokinesis is complete, we're going to head back into the interphase phase of life. So lastly, as we said, cytokinesis is kind of our final part, and this is where we get the full division of all of the cytoplasm, which includes the organelles and the cytosol, so that it's fairly evenly distributed between the two daughter cells. So two daughter cells at this point are going to have the correct number of chromosomes identical to the parent cell, and then also going to have all of the cytoplasm, etc. They need to continue into interphase. So note that the division of the nucleus in mitosis and the division of the cytoplasm, cytokinesis, are separate events, but in real life, things aren't super clear cut. And that's why sometimes you'll see like, oh, we're in early telophase, or we're in late telophase, or we're in early prophase, or whatever, because obviously it's not like things just happen superstructurally and we can like time it perfectly. A lot of these phases kind of overlap with one another. So cytokinesis, which is the division of that material into the two cells, can actually start as early as anaphase, but usually will start during that last phase, during telophase. So that in a nutshell is mitosis, kiddos.